All right, so this is a time for just general sort of research questions meant for our speakers and other things we can talk about more in a kind of discussion. So we welcome any questions you have. And I think there was one that I wasn't asked kind of during the little switchover break here about dosing for toxicity versus dosing for efficacy. And again, I think it's important to reiterate that safety is the primary goal here. So we want to make sure that we're giving a drug that is not doing any harm before we think about whether it's doing any good. So that is the primary concern. We should have a much better idea of how this ASO is behaving after these IND enabling toxicity studies, and that's going to help determine what that starting dose might look like. Yeah. All right. Are there any other questions to start us off? Ganesh?
Yeah, is there much overlap? If there's much overlap between what you're doing, Dr. Padia, and Lorem? So is there much overlap, especially related to, in relation to mice and rat safety studies between your lab and Lorem?
So I think so the ASO work that I showed is all done in collaboration with Ionis. So they helped us screen the ASOs, they identified the ASOs in cell culture, and we've injected them in mice, right? They are targeted to the human gene, but it's important to remember that in the mice, it's only the human cDNA, right? Whereas most likely the ASOs that are being used in the human would probably be targeted to the intronic region because apparently those are more effective. So I would say that, they are different. They're two separate kind of things. And the stuff that we are looking at clearly is not toxic because we've injected it into the brain. Some were toxic, some were not, but they're probably going to be different ASOs. But I think they're very complementary because I think if we can continue our work and show that the ASOs are therapeutically effective, that gives a huge boost to, the stuff that's kind of being done. So.
Yeah, and the ASOs, so Ionis and Lorem are obviously attached at the hip, especially earlier on when this program started. And I do believe that Ionis was doing much of the design for the ASOs during that time, and they designed the ASOs that we're studying and developing for the patients. But yeah, the screening involved all of the gene, including the intronic regions. So it's unlikely that they're the exact same sequences. And it's also unlikely that they would share the lead candidates for external research. So they want to make sure that those are really done under their watchful eye for each of those experiments.
I read the question here. Can I read it? From Ujjal.
Sure, go ahead.
So the question was about how do we create how do we avail the community's resources to develop a unified theory of ALD pathogenicity? What is the current consensus on the cause-effect relationship between different types of cells, organocytes, astrocytes, etc., and how this causes downstream demyelination? What experiment studies would you propose that would allow us to tie various research streams together? So that's a big question, Joe. So I will start, but I think we all need to, let's say, talk a lot about this because I think that one of the key aspects of this meeting is that it's also important that there are still different ideas about the disease. It's something very important because if we all add the same, let's say, hypotheses or also models, it would be impossible to really do something good. And I think we will be able to unify everything, let's say, we three and everybody together once we'll know something more. And to know something more, we have to continue, like, for example, the questions that were raised about the vascularization, question about downstream pathways, demyelination. Is it before the astrocytes or after? Is it the cause of the effect? We are all trying really to understand, and I think that actually we're on the right path, all of us. And I think that the key is not a specific experiments now. I would say, of course, creating engineer mouse models with a human brain that has ADLD different phenotypes, even the one without the pathological phenotype. So create the best models, but the time for creating a model is so long, and the cells of ADLD are so sufferant that make our effort even more challenging. So every model we try to develop, I guess, is similar for Quasar. It's hard because of the sufferance of the cells. So no specific experiments. I think that the best way is to continue this, so to talk and share our data. And for sure, once any of our model be more reliable, then we have to collect the data for all of us.
I don't know what to add because it's a very big question and multiple components to it. So I would just say, yes, it's tough to come up with one specific experiment or a single experiment that's going to answer everything. And I think you just have to move forward and come up, and I think it would be simplistic to think that, this disease there's just one pathway and that's what's causing. Huntington's disease has been known for the last nearly 40 years. We still don't know the exact pathways that are responsible for Huntington's disease. But that does not prevent you from developing therapies, or trying out different targeted therapies. So I think maybe there are multiple pathways involved. Maybe there are multiple cell types involved. And I think it's important to continue, like Stefano was saying, trying different models. And, understand disease mechanism is one thing, but also kind of at the same time, parallelly, you don't have to 100% understand a disease mechanism before you start looking at therapy, because we do have a target. And I think, at least some of the reversibility stuff we've shown suggests that Lamin B1 is a good target. So I think that would be worthwhile. And so I think they are parallel, they are complementary kind of approaches, but they do not have to be kind of exclusive, and you don't have to wait for one to do the other.
And I'm also really interested to see if there are areas where, we're going to layer in therapies, right? So we can maybe knock down Lamin B1 expression with an ASO, or maybe in the future some other modality. But then how do we, encourage remyelination? How do we fix some of the other defects that have taken place already that we can't go back and fix just by waiting? That's not going to happen. So how do we recover more normal function once we, kind of take our foot off the gas of Lamin B1 expression?
And that's also why I think at the end, even for future therapies, there will be a combined therapy also for that, as combined studies there are. And if you think about the phenotypes, they are so different. It will be so hard to detect patients. So we have to be sure which one we can diagnose with a disease that is effectively affecting the patient. So because if we have a duplication that has a normal phenotype, we have to be sure that that cannot be a patient we want to treat. So we really understand which are the targets also apart from Lamin B1, because there could be other easier targets. But the main question is also when. That's always tricky part. When would you start to treat the patient? When would you have the first diagnosis and be sure that is this kind of patient? So many questions, unfortunately, are still open, but I agree with both Margo and Kazar saying that you have to work in parallel with both trying therapies, testing therapies, and developing models and understanding mechanisms.
I'm also curious if either of you have looked at any genetic modifiers or other modifiers of phenotypes within either cell systems or in patients, especially as we get farther and farther into specific pathways and you're looking at patient individual cell responses and phenotypes. Can we correlate that back to anything we might find in a genome that might also predict? And is it not necessarily a downstream effect maybe of ADLD, but another, genetic modifier of that response?
I think your earlier question to Riley about, is there interfamilial variability in disease progression was a really important one, because I think that will really answer, that, right? Because if you assume that the mutation is the same in patients and patients have, wildly different disease progressions, then I think the modifier would be really interesting. And I think that would be really interesting. But I think that's also complicated because, like Riley was saying, all of us are different, right? And because this is such an ultra rare disease, you would have to pick up pretty much everybody and sequence their genome. So I don't know. It's a really important question. I think it is definitely relevant. It's kind of tough to do. I think that's my only thought.
I agree. On the last sentence specifically, it is very tough to do it. And especially trying to understand from what comes what, it's always a challenge in any kind of model, also from the patients. I think that once we'll have the RNA-seq data of the cells of a lot of patients, we'll have a bit more of information about that.
Another significant challenge with an adult onset disorder is what else, right? Thinking about our fearless sim leader and thinking about the exposome and what else can modify. It'd be interesting to know, do the phenotypic variability in patients, is that also reflected in any cell responses or cell phenotypes? Can we explain that through a different mechanism? But lots of questions. I think they're going to take some time to answer.
Well, I think that one thing that was said at the beginning by Riley that I really agree with is that we always think that some of rare diseases are rare diseases, but then we discover that we could discover they're not that rare. Or even what I'm starting to see is that in the past, we used to learn from more diffuse disease information for rare disease. What's going on now, it's kind of an opposite trend. So the research is going so in detail for rare diseases that I understand we are now finding information that could be useful for Parkinson, Alzheimer, and other diseases because some mechanisms could be very common. So exposome and so on opens a big world of research, and I agree because a lot of informations now are really interdisciplinary connected between different diseases. So I think we also have to remain open, as we are doing with this Congress, that I can see you invited different experts and different people, not only ADLD. I think that's also a very good thing because we can really copy or have ideas for other disease and give ideas to other researchers that are doing other diseases. So I think this also would be a very important exchange.
I think one question I had, and maybe this is more relevant for the clinically themed talks, is how a kind of clinical trial, hopefully assuming that, the ASOs look really great in terms of safety, how a clinical trial would be kind of conducted. And I think, obviously a great example is Huntington's. Very similar. It's adult onset, potentially if you have low repeat numbers, long progression. Does anybody have any idea how the Huntington's kind of trial, what did they use as, outcome measures? What did they use as target kind of engagement?
I don't know that I can speak intelligently on that.
I mean, because I think, clearly you cannot do a 20-year clinical trial, right? So we have to have some kind of biomarker or something. And I'm wondering if they use the same kind of strategy for Huntington's.
The biomarker is going to be a big question. And we know that for like tofersen, it actually didn't meet the criteria for their endpoint analysis. But because they had the neurofilament light chain, and that's been shown in several other ASO trials for neurodegenerative diseases, it got approval anyway. Summary, right, brief. So if we can get to that level for ADLD, I think it would give us a lot of power to have an objective marker of disease progression. But again, we have so few patients. It's going to take a lot of work, I think, to get coordinated to make sure we're getting samples, that we're doing some of that biomarker development and that, granular phenotyping, thinking about, a trial, right? We know that maybe all of this clinical testing or evaluations may not change their course of disease if there is no therapy. So it's tough to get that data without having a study to capture it. So I think really initiating some of that work as soon as we can will hopefully allow us, when we know something about this ASO we're starting soon, or other therapies that hopefully are coming online, and drug repurposing or others, as soon as we get to a trial, we'll have a little bit more data to know what to do and how to do it. I think along with that is the patient stratification. So understanding for sure the differences in phenotypes between deletions and duplications. And if we can predict what that variability looks like, or if there's, any markers of, or predicting factors that can, predicting factors of disease trajectory, whether one is more, steep than another, is going to be really important, because we can't exclude a patient. We have so few that we can't say we're only going to take these patients with exactly this duplication or exactly this onset, because we'll be out of patients. So we have to be as general as we can, which means we need to understand how to stratify them to measure benefit across the board, given their variability. So lots of things to do.
Actually, one comment related to that is, everybody says, oh yeah, there are so few patients, I don't think that's the case. I think the more and more, because I'm getting more and more inquiries every day. There was one case where this is somebody who was misdiagnosed as, I think, a CSF1R related leukodystrophy. And it turns out it was ADLD. So I have a feeling that this is grossly underdiagnosed, and there are going to be many, many more patients than we kind of expect. And, I don't think that should change our focus on it. Even if there are 10 patients, still a fatal disease. But I think there are many, many more patients than we expect, especially because if you look at the age of onset, it's later, it's like 40, 50, right? All of them have kids before they're kind of born. Nobody's doing pre-implantation genetic diagnosis. So there has to be a large number, I think there's a pretty large number of patients.
Yeah, I think that's an excellent point. I agree. There are likely a lot more. And then that also gets at the question of, I'm maybe getting into the translational and clinical side a little bit here too, but, as a decentralized trial, how do we get patients where they need to go? Mobility is a significant issue, right? There's lots of concerns, but yeah, I agree. Likely way underdiagnosed. All right, Stefano, any other closing comments? Otherwise, I think maybe we'll wrap
The only
this
thing
discussion.
I'm happy to say is to thank you, and thank all the people in presence there. And I found this first part already very interesting. Not talking about my talk, your talks, and nothing. Looking forward for the next part.